In this lesson, we continue our look at neurons and neurophysiology by investigating action potentials in more detail, and here are your lesson objectives. Over on the right here, I have the same two membrane proteins that we saw in the previous lesson. I'm going to again draw a membrane here, and up at the top is going to be outside of the neuron, and down at the bottom is going to be inside the neuron, where we've seen we have a negative resting membrane potential. And the protein on the left here is meant to be a ligand-gated ion channel, so that when a ligand, in this case a neurotransmitter, NT for neurotransmitter, binds, it opens and allows ions to move down their electrochemical gradient. As we've seen, an example of that is sodium moving into the neuron down its electrochemical gradient. And these ligand-gated ion channels that respond to neurotransmitter are on the dendrites and cell body of this neuron throughout, and they're receiving neurotransmitter from the neurons one step back. So although only one neuron is drawn here, there are many neurons synapsing on it. There are many, but I'm just going to draw a representative few here, and they're releasing neurotransmitter that is then binding to these ligand-gated ion channels and opening them. If sodium enters the neuron, that's going to depolarize the neuron. If you imagine time on the x-axis and membrane potential on the y-axis here, that's going to create a positive bump in the membrane potential. We're going to call that a positive graded potential or an excitatory potential because it makes this neuron more likely to fire. In contrast, if the ligand-gated ion channel lets a negative ion like chloride in, then that's going to hyperpolarize the membrane potential, make it more negative, and because that takes it further away from the threshold potential that we saw in the previous lesson, this is an inhibitory potential. It makes the neuron less likely to fire. Now those signals are coming into the dendrites in the cell body all over. There's neurotransmitter being released at many points here onto this neuron from previous neurons, and they are going to add to a total membrane potential. Some positive, perhaps some negative signals coming in at all of these different places, and they add over both time and space. We call that temporal summation and spatial summation. And so if you have multiple positive graded potentials, they can actually add like this and bring the membrane potential higher and higher, closer to the threshold potential. Let's look down here at what that looks like. This is meant to be an oscilloscope tracing. We've seen that the resting membrane potential is about negative 70 millivolts. And if you get multiple excitatory potentials, you can sum to get the membrane potential closer and closer to threshold potential, which, as we said in the previous lesson, is typically around negative 55 millivolts. And at that potential, something very special happens. At that potential, voltage-gated sodium channels open. So this is meant to be a voltage-gated sodium channel, and they open in response to depolarization. When the membrane depolarizes to threshold potential, they open and allow sodium in. So this gate here swings open like this, and sodium rushes in. Now that's a positive feedback cycle because depolarization is causing more sodium influx and therefore more depolarization. On the oscilloscope tracing, that causes a big positive spike as sodium rushes in through those voltage-gated sodium channels. Now there are also voltage-gated potassium channels. They open at the same time, but they are a little bit slower. After a little bit of time, the second gate here, called the inactivation gate on the voltage-gated sodium channels, closes. That stops that positive feedback cycle, and together with the opening of the voltage-gated potassium channels, that causes the membrane potential to drop back down like this. So this is potassium now going out from the inside to the outside of the neuron down its electrochemical gradient, and the membrane potential actually hyperpolarizes past resting membrane potential. It overshoots and then comes back to resting membrane potential as a result of this potassium movement out. Now the interplay of these different membrane proteins and the gates in them gives rise to a phenomenon known as the refractory period, which is a period during which the segment of axon either can't fire another action potential or can only do so with an extra big stimulus. So the inactivation gates that we saw above, the little ball and chain, close up the sodium channels after an action potential fires, and they take a little while to reset. And while those are closed, that segment of the axon just can't fire another action potential, and we call that the absolute refractory period. No matter how big the stimulus is, those inactivation gates in the sodium channels, the voltage-gated sodium channels, are closed, and so no more action potentials can fire. That means that there's a limited frequency at which action potentials can fire. They can't get spaced too close together. There is also a relative refractory period, which is longer and follows from the absolute refractory period, and that's when some of those inactivation gates in a given segment of neuron are closed, and also because of this hyperpolarization, those two effects together mean that you actually could fire another action potential, but it takes an extra big stimulus to do so. If we look up here, this is meant to represent an axon shown from the side. It has a negative resting membrane potential, so it's negative inside and relatively positive outside. If we imagine an action potential moving through this axon from left to right, what's happening is that sodium is moving in as the voltage-gated sodium channels open, and that's making this segment positive, bringing it to threshold, and so more sodium is moving in. And the movement of sodium ions and positive charge this way inside the neuron is bringing each new segment to threshold. These movements inside the neuron of the positive charges are called local currents. They're what are depolarizing the next segment and bringing the membrane to threshold, opening the voltage-gated sodium channels. So we have this kind of wave effect, very much like the kind of wave you would do at a sports stadium, where the positive charges are causing the influx of positive charges at the next section. So that's how the action potential travels like a wave down the axon. To illustrate, here's a simulation from PhET. The URL is on the screen now, and we see a neuron coming towards us, and at the bottom we have membrane potential versus time. If I press play, we're going to see the action potential moving towards us as this glowing line, and I'm now going to zoom in so we can watch what's happening at this segment. So the segments before, or into the screen, have already depolarized. Sodium has moved through voltage-gated sodium channels and made that part of the membrane positive. That's now depolarized this part of the membrane, and if I scrub through here in slow motion, we'll see as the depolarization arrives here, this first channel here on the left, which is in red, this is a voltage-gated sodium channel, and it's going to open in a moment, and this one is a voltage-gated potassium channel. It's going to open too, but a little bit more slowly. So as the depolarization arrives here, we see the voltage-gated sodium channel open and sodium is moving in. And this is the inactivation gate that's going to swing closed in a moment. The potassium channels are opening too, but more slowly. As we keep moving forwards here slowly in time, we see potassium starting to leave. And then the inactivation gate on the voltage-gated sodium channels closes, stops the influx of sodium, but the efflux, or moving out of potassium, is still happening. That actually causes the membrane potential to overshoot a little bit here. And in the blue and the green, we see leak channels, which we're not going to focus on here, but are helping to maintain the resting membrane potential and reset it. If I go back again here and play that in slow motion, let's watch one more time as the voltage-gated sodium and potassium channels open. And this also illustrates where the refractory period comes from, as this inactivation gate closes the sodium channels and the neuron can't fire in the segment until it opens again, like this. Now let's finish by thinking about how myelination speeds conduction. Because all of this movement of ions takes a little bit of time. As we saw in an earlier lesson, Schwann cells wrap around the neurons, so here I'm showing this in cross-section. And that does two things. First, it insulates the neuron so that electrical current can't leak out, but it also limits the number of places where you can actually have ion channels. There are no ion channels in this part under the Schwann cells, and so ions are only moving at the nodes of Ranvier, here and here. And electrically, that's observed as the action potential jumping from one node to the next. At each node, it is getting replenished by sodium moving in, but because that only has to happen at the nodes and not in these segments under the Schwann cells, that saves time because ion movement takes time. So let's look at action potentials and the ion movements that underlie them.